dorsiventral region of that is dorsiventrally differentiated Marchantia thallus we have studied. We know about the morphology and anatomy. Now we'll proceed to the reproduction. Reproduction in Marchantia. These are the lower plants. So they reproduce by vegetative method as well as sexual method. So vegetative reproduction now. It is by means of, that is, by progressive death and decay of the cells it takes place. That is by fragmentation. Some of the fragments in the lower diagram you're seeing right hand side, some pieces which may be broken down, mature cells, then that may be broken down. By chance, if it is broken down, it can develop into a new plant body of Marchantia. So mature cells are present at the posterior end and they are short-lived. New cells are regularly formed from apical cells. Death and decay, therefore, naturally starts from the posterior end. As the process of decay reaches the point of dichotomy, two apical part of the thallus separates. Each grows into a new plant. As you know, at the notch, meristematic tissue, the growing tissue is present. So that fragment, if it is broken, that can develop into a new plant. So reproduction, not only by fragmentation, another method is by means of adventitious branches. Here, adventitious branches you're seeing on in the diagram you're seeing on the lower side. Adventitious branches means the branches which are growing not from the normal position, extra branches which are rising. So some species possess adventitious branches on the ventral surface. By chance, these adventitious branches, if they are separated, they'll grow into a new thallus of Marchantia. The special feature of Marchantia is it reproduces by another method of vegetative reproduction, that is by means of gamma cups. Questions can be asked on this mostly, that is gamma cups. In the first diagram you studied the gamma cups on the upper side, that is the dorsal side. In the midrib region they'll have gamma cups, so inside which gamma produce, this gamma may help in reproduction, that is vegetative reproduction. So about the gamma. Gamma is singular, gamma plural. Gamma cups are capsules present on the dorsal surface along midrib region. So this already we have seen. Margins are hyaline. You've seen the cup, that is gamma cup is hyaline, that is colorless. Hyaline means colorless. It is lobed. It will have lobes. It is not just a cup-like structure, but it will, with the lobes it is. And it is spiny and entire. From the floor of gamma cup-like structure on the middle, many small stalked, discoid and biconvex gamma will arise. Look at the diagram on the upper side diagram. There you are seeing stalked gamma. The cup-like structure you are seeing inside the cup-like structure, somewhat dumbbell shaped with a notch at the middle. Actually, gamma are somewhat dumbbell shaped, and each gamma, there is so many gamma are present. Each gamma is attached to the floor of the cup by means of a stalk, that is called a stalk of the gamma. Gamma is constricted in the middle and has two notches. As I told on the lower diagram, you can see each gamma will have a constriction at the middle, that is called as notch. It possesses, at that notch, two notches possesses a row of apical cells. At that notch, you will see apical cells. These cells, they help in cell division. Gamma contains chloroplast containing cells and rhizoidal cells. In the gamma lower side diagram, some of the larger cells you are seeing, some of the cells they'll store oil cells you are seeing. In addition to the oil cells, some of the cells they'll have chloroplast inside that. It helps in photosynthesis, as well as rhizoidal cells are also present, which will in future it will give rise to the rhizoids. Some in between the gamma cups, mucilage hair-like structures are present. Some mucilage hairs also arise from gamma cups floor, which imbibes water and help in dispersal of gamma. So whenever water is available during rainy season, in the gamma cup, it will be filled with water, and this mucilaginous hair, they'll imbibe, they'll absorb the water, and that will be retained inside this. So when the mature gamma are dispersed from the gamma cup, that time this water helps in dispersal. Gamma, in coming with the ground or the substratum, it develops into a new plant. Each gamma can give rise to one or more Marchantia plants. Then about the sexual reproduction. Vegetative reproduction, it takes place by fragmentation, adventitious buds, as well as by gamma cups. Sexual reproduction next. Sexual reproduction is very, very special in Marchantia. Marchantia is a heterothallic or dioecious plant. What is heterothallic? Heterothallic means anyhow dorsiventral differentiated will be there, as well as male and female plants are different. So that is dioecious plant or heterothallic. Male plant and female plant are both are different. Male thallus, it bears antheridiophore. Phore, Phore, phore means the stalk. Stalk on which antheridia are produced, that is called as antheridiophore. So male thallus bears antheridiophores and female thallus it bears archegoniophores. They arise, both antheridia and archegoniophores. They arise from the distal end of the thallus from growing point. Growing point, that is the notches, and after their formation, they grow into a, that is growth of the thallus is ceased, means somewhat growth of the thallus is reduced, it stops. Once the antheridia and archegonia are formed, then the normal vegetative growth of the thallus is reduced. Here you are seeing the antheridiophore. Antheridiophore is, it will produce antheridia, that is male reproductive part. So here you are seeing antheridiophore in the diagram. Once you look at the diagram, green thallus, dichotomously branched thallus. At the tip of the thallus, you are seeing a elongated stalk with a stalk-like structure. So this stalk is antheridiophore. At the tip of the antheridiophore, you are seeing a cup-like with many lobes. So in which antheridia are produced. Antheridiophore, it has 1 to 3 centimeter long. The length of this antheridiophore is 1 to 3 centimeters long stalk that bears 8-lobed peltate disc, means a plate-like disc is present with the lobes. 8 lobes are present, as you are seeing in the diagram. Inner structure is similar to the thallus. If you take the tears of this, it is similar to the thallus of Marchantia, with upper epidermis and with air pores and upper side photosynthetic filaments, all that. Then antheridial chamber. Antheridial chamber alternates with air chambers. So in the thallus, you'll see the antheridial chambers and alternating with air chambers. Now about the antheridial chamber. Each antheridial chamber, it contains a single antheridia and opens externally by a pore called ostiole. Ostiole means always it is the opening. On each lobe, antheridia arise acropetally. Acropetally means older one at the periphery and at the center, younger ones are present. That is acropetalous succession. Antheridiophore has two longitudinal grooves. Each antheridiophore in the diagram, next to diagram, you can see that. Okay, it's not there. Then anyhow, antheridiophore has two longitudinal grooves. About each antheridia, in each antheridial chamber, one antheridia is present. In the diagram, you're seeing the antheridia. Each antheridia in Marchantia is club-shaped. In all bryophytes, it is club-shaped antheridium. Club అంటే గద ఆకారంలో ఉంటుంది. So club-shaped antheridium you're seeing. A mature antheridium is globular or oval with multicellular stalk. So this complete stalk and the upper part, globular or oval shaped part, it is called as antheridium. That is club shaped antheridium. CLUB club. The body of antheridium has a single layer, that is the jacket layer it is called, and enclosing inside it, and so many cells you are seeing, the partitions. Each partition is that will develop into a androcytes. Androcytes, which eventually metamorphoses into anthrozoids. So each one that will develop into a metamorphosis means afterwards it will undergo change and it develops into a anthrozoid, which is biflagellate. Then after maturation, after the complete development of the antheridium, the wall will rupture, anthrozoids will come out. That is the dehiscence of antheridium. Water, in the presence of water only, anthrozoids can swim, biflagellate anthrozoids, so water is required. When the water enters the antheridial chamber through ostiole, then by the time antheridia will be mature and this antheridia released. In along with water, they'll come out through the ostioles. They are dispersed in water and they'll reach the archegonia. Some cells at the distal end disintegrate. That is the antheridial wall you are seeing. Usually the terminal cells or the outermost cells of this, the wall, jacket wall, they'll disintegrate, and when they come intact water and antheridium is ruptured and anthrozoids, they'll come out. About the archegoniophore. Archegoniophore is formed on female Marchantia plant body. So here in the diagram, upper diagram, you're seeing that is Marchantia plant body with lobes. One of the lobes on the terminal part near the notch, you are seeing a stalk-like structure that is called as archegoniophore. At the tip of the archegoniophore, many lobed hanging structures you are seeing, which is somewhat umbrella-like structure, that is archegoniophore, whereas antheridiophore is disc-like structure. So about the archegoniophore, it has slightly longer stalk, that is 2 to 5 centimeters, and a terminal disc with here also 8 lobes are present. Archegonia are born on the dorsal surface in acropetalous succession. On the upper side of the thallus only they are born, and they are here also they are produced in acropetalous succession. Internal structure of this archegoniophore is also similar to the normal thallus, that with the upper epidermis, pores, air pores, and air chambers are present, photosynthetic region, all that are present. Here in the lower diagram, you are seeing that is archegoniophore lobes you are seeing. Structure of archegonium. In bryophytes, archegonium is flask shaped. We'll call it as flask shaped. So about the archegonium you're seeing in the diagram, you're seeing it is inverted position. So this is not straight. This is inverted. The archegonium is stalked flask shaped structure with a basal swollen venter. Upper side you're seeing the venter and a elongated neck which is facing down. The venter is surrounded by one-celled thick sterile jackets. The outer wall is called a jacket wall. It contains at the center a large egg cell and relatively smaller venter canal cells. Violet colored structure in the venter region, you're seeing somewhat swollen region. There you're seeing the venter, that is egg cell. Just above the egg cell, venter canal cells will be there. In the neck, here you are unable to see, but that is the external structure only is shown. But neck it will have a canal, and that is it consists of neck canal cells. The neck consists of 6 vertical rows of cells. That outer wall 6 rows called neck cells, which encloses, that is inside this 4 to 8 neck canal cells are present. So also at the tip of the neck, this is not always open. It is covered by 4 cover cells. Lowermost part, that is the cover cells are present. Usually there are 4. Now about the fertilization. Fertilization here, that is nothing but fusion of the male and female gametes. In the diagram, you're seeing left hand side, that is archegoniophore. Right hand side, you're seeing the antheridiophore. Antheridiophore in the antheridial chamber, antherid it is, and each antheridia will produce the antherozoids. Antherozoid, they'll swim in water and somehow they'll reach the archegonia. There in the, that is in the venter, egg cell is present. There it fuses, thus fertilization will takes place. So about that you are studying here. What is essential for fertilization? Archegonia placed upright on archegonial disc. Right hand side diagram. Ventral canal cells and neck canals, left hand side diagram. Ventral canal cell and neck canal cells disintegrate. After the maturation of the archegonia, in the venter canal cell will be there above the egg cell. So that will disintegrate, as well as neck canal cells which are present in the neck region, they'll also disintegrate to form a mucilaginous substance which helps in absorb, that is imbibe water, and results in separation of cover cells by pressure from the inner cell by the disintegration of the venter cells and the venter cell and the neck canal cells, NCC they are called. Due to that, pressure is created a mucilaginous substance. It comes out because it imbibes water, and due to that, pressure is created. Cover cells will break, and it makes a opening or canal for the easy entry of antherozoids. Antherozoids present on the surface of the antheridial disc are flashed by raindrops on the surface of archegonial disc. They are attracted chemotactically towards the neck of the archegonium. This archegonium, by the disintegration, it produces a mucilaginous substance, which will produce some chemicals. So there is chemotactic movement. So we'll say, so due to that, antherozoids are attracted towards the archegonia. neck of the archegonia, and one of the anthrozoid, it enters the archegonium, and then it fuses with the egg cell to form the zygote. After fertilization, there is a post fertilization, some changes will be there in Marchantia. Here mainly what you are seeing is inversion of archegonia. Now archegonia, in each archegonia, that is egg cell it fertilized, and after that the antheridiophore, the structure, the lobes which are present there, they are inverted. After fertilization, archegoniophore elongates, accomplished by rapid growth of central part of a archegonial disc. That leads to the inversion of archegonia. In the upper diagram, you're seeing first diagram, archegonia straight, but after fertilization, they'll bend down. The second diagram, bending structure you're seeing, inverted structure. The arrangement of archegonia is also reversed from acropetalous to basipetalous. So here the arrangement, usually they are acropetalous succession, but after, due to inversion, that is inversion, it takes the basipetalous succession. There is the older one at the center and younger one toward the periphery. That is archegonia. This process of inversion is accomplished by development of one cell thick tissue called as pericytium. This is important to protect they're hanging down, archegonia hanging down. To protect that, a thin layer is formed that is called as pericytium, or it is also known as involucre. On the both the side of each archegonial row, they are formed. In the lower diagram C and D, you are seeing some thin structures. They are covering the archegonia, fertilized archegonia, that is called as pericytium. Also, long green finger-like projections are formed from the outer layer of this. Next, after fertilization, the zygote is formed. That zygote develops into embryo. Zygote always develops into embryo. Zygote is diploid, and embryo, it contains the meristematic cells. They are all the cells are diploid. These zygote afterwards, that is the embryo, afterwards it will differentiated into foot, seta, and capsule. In the diagram you're seeing, foot is present upside because they are hanging down. The sporophyte is hanging down. That is the specialty of Marchantia. Foot is present upper side. Then you're seeing the seta and lower hanging structure, that is the capsular region. So zygote develops to form sporophyte. Zygote is diploid. Sporophyte is also with 2n, that is diploid cells. It is differentiated foot, seta, and capsule. Foot, it is a basal bulbous part which anchors and provides nutrition to the sporophyte. In this diagram, you're seeing the foot is present above. That is the archegonia region, the venter of the archegonia. There, foot you're seeing. Foot, it is helps in attachment, encouragement. It provides the nutrition also to the developing sporophyte. Above that means here in the diagram, lower side of the foot, you're seeing the seta. Seta is somewhat short. It is the stalk that connects foot to the capsule. Just it helps in attachment only. Capsule. Capsule is yellow colored usually. Broad structure you're seeing which is hanging down. That is a capsule. In the diagram, you can identify that capsule. It is yellow colored, oval structure, and has a single layer jacket layer. That is jacket wall is present. In young sporophyte, capsule encloses sporogenous mass. Inside the capsule, the outer, that is a jacket wall, capsule's jacket wall, So many cells are present. These cells are called as sporogenous cells. There is a sporogenous mass which differentiate into spore mother cells. And some of the cells will differentiate to elater mother cells. Elater mother cells. These elater mother cells will develop into the elaters after reduction division, whereas spore mother cells after reduction division it produces the tetrads of spores. You're in the right hand side, you're seeing the elongated fiber like structure. They are the elaters. So they'll have reticulate thickening on that you're seeing. And the lower side, you're seeing round structures. These are the spores, which are haploid, which are formed after reduction division. Spore mother cells, as I told, it gave rise to the spore tetrads by meiosis, and elater mother cells will give rise to the elaters, which are diploid and sterile. They'll not help in fertilization. They are diploid and they are sterile. During the development of sporophyte from zygote, the venter wall gave rise to a 2 to 3 layered protective covering of sporophyte. It is called as calyptra. So that is the speciality here, because these are hanging down this sporophyte. To protect the sporophyte, extra layers are formed from the venter region of the archegonia, and that protective layer, yes, that is called as calyptra. In the diagram, you're seeing beside the capsule, one layer you're seeing on both the sides, that is calyptra. Not only calyptra, surrounding that and some other layers are present. The basal cells of the archegonium give rise to the one-celled thick collar-like outgrowths outside the calyptra. These are called as perigonium. Perigonium, or that is called as pseudoperianth. It is a pseudoperianth, or it is also known as perigonium or perigonium, that is. Then towards maturity of the sporophyte. When sporophyte is completely mature, the seta elongates. The seta part above the foot, you are seeing the seta. In between the foot and that is capsule, seta is present. So that will suddenly elongates and pushes the mature capsule out through the calyptra, perigonium, and pericardium layers. So it comes out. And afterwards, the outer wall of the capsule, the jacket wall of the capsule, it breaks, and so here you are seeing the dehiscence of sporophyte. Sporophyte is broken down. Here, erect position it is shown. Actually, it is inverted. The wall layer will break. The outer wall layer will break into many parts, and spores are released. Single layered capsule walls splits into a number of longitudinal valves. Here you are seeing the valves, which extends from apex toward the middle of the capsule. So normal fruit capsules, how it breaks, that's why that like that here also capsule is broken down into many 4 or 5 layers, that is parts, valves, and due to that spores are released. The valves are rolled back due to the annular thickening in jacket layer. So they fold back. Jerky movement of the elaters due to their hygroscopic nature leads to the loosening of spore mass, and scattering of spores in the air. Here, special characters of the elaters you have to remember. Elaters are hygroscopic, means they'll absorb the water, and due to that it will put pressure on the spores. So along with the elater, spores are released in groups, in masses in the air. They are spread, they are scattered in the air. After, when once the spores are dispersed in the air, they'll find a suitable place on the earth and they will germinate. So, germination of the spores. Spores are haploid. Spores are available for about 1 year. Means survival period is up to 1 year also they'll survive till they get the suitable conditions, moisture, suitable substratum, that is humidity, all this. Till that, for up to 1 year also they can survive. Under favorable conditions, they absorb moisture from the substratum and increase in size. In the spores, chloroplasts are present. So chloroplast reappears at this stage. Chloroplast helps in photosynthesis and they'll produce the food materials. Spores undergo repeated division, that is mitotic division, to form 6 to 8 celled filamentous structure with a rhizoid at the end. In the diagram, you can see rhizoids. In the first diagram, you are seeing the left hand side, a spore. Then next diagram, you are seeing it is 2-celled. Next diagram, it will be 4-celled. Then 8-celled, like that it divides 6 to 8 times and it will produce 6 to 8 filamentous structure. Some of the cells will give rise to the rhizoids towards one side, which helps in anchorage, or that is fixation, to absorb the water. Meanwhile, the upper cells will produce, that is, they'll regain the chloroplast, they help in photosynthesis. The apical cell cuts off derivatives on the lateral side and finally it gives rise to the gametophyte. So it is the protonema-like structure. This protonema will give rise to the spores will germinate to produce the protonema. Protonema will produce the gametophytic plant body, that is the thallus of Marchantia. So with this life cycle of Marchantia is finished. Life cycle here is alternation of generation is seen. Heteromorphic alternation of generation. Sporophyte alternates with gametophyte. Plant body is a gametophyte. Gametophyte will produce the respective gametangia. Male and female plants are different. This female plant, after fertilization, it produces the zygote. Zygote will produce the embryo. Embryo will produce that is the sporophyte. Sporophyte, capsularization, spore mother cells undergo reduction division to produce the tetrads of spores. Spores are haploid. Again, they germinate and produces the plant body of Marchantia, gametophyte. Do you have any doubts?
No, madam. Crystal clear, madam.
Is
No, madam.
it
No,
clear?
madam. Clear,
Oh,
madam.
okay.
Thank you, madam.
Thank you. Thank you. Anybody else? Any doubt?
Okay, madam, thank you very much. It's a crystal clear class regarding of our hepatic apsida type study Marchantia, morphology, anatomy, reproduction, sexual reproduction, especially the sporophytic structure and the capsule of the Marchantia is clear explained about the Marchantia type study. It is very useful for the not only for UG and PG students. It is also very helpful to all the competitive aspirants. Thank you very much, madam. Tomorrow we will meet same time.
Okay.